So I'm here to present and represent an industry perspective. And certainly there has been a tremendous amount of excitement in the field of CH activation, CH functionalization. And the challenge for us in industry is how can we harness all of this energy, all of this enthusiasm, and translate that into meaningful impacts into how we actually discover drugs in this case. And so I'm going to give you a few case studies for how we've been using CH functionalization chemistry and some actual examples from some medchem programs here at Merck. So first of all, for those who aren't as familiar with medchem, basically it's a simple design cycle. One has some particular hypothesis around biology, certain molecules that could interact with the biological system in a therapeutic manner. Design molecules, and of course you need to make those molecules, and then you can test them and see if your hypothesis was correct. You can then evolve your hypothesis and hopefully work your way toward a drug. And there's a lot of sophisticated design that goes into this, and of course synthesis is a big component. And there are a lot of themes that came out in Jin's talk, that I want to re-emphasize, one of which is speed. So it's very important that one goes around this design cycle as quickly as possible. And of course, synthetic chemistry is key to that. One way we say this at Merck is that synthetic chemistry enables design. You want to be able to make every molecule that you design. You don't want to be constrained in the molecules you design based on what you think you can make. And so that's very important that we can go around this cycle with high success and high speed. But beyond that, synthetic chemistry can even inspire design. These new tools that Jin and that people in the center, people in this field are inventing can actually give us access to new kinds of molecules that perhaps we hadn't even thought of making before. And then that can open up new therapeutic opportunities for us in terms of inventing new drugs. So both of these concepts are very powerful. And so that brings us to CH functionalization chemistry and this concept of late-stage functionalization, or as Jin was calling it, molecular editing, a very powerful concept that, of course, really wasn't even on anybody's radar screens 10 years ago. But with the advent of these new chemistries, these new tools, this is becoming something that's more and more realistic. So if we take a drug such as voriconazole, and we've grown accustomed to not drawing the CH bonds by and large, but if we can start to think of these CH bonds as functional groups, as potential points of diversification, then we have a lot of possibilities that open up to us for editing a molecule like this to really tune in desired properties. So there are a lot of exciting potential applications of late-stage functionalization. Basically, one can imagine addressing any of the commonly encountered medicinal chemistry problems through this type of approach. The challenges are, again, to quote Jin, is around regioselectivity, and I would also add in functional group compatibility. A lot of these chemistries have just been invented and perhaps have never been tried on a molecule as complex as voriconazole. The other challenge for people in industry, other than that, perhaps we don't grasp all of the mechanistic subtleties of some of these catalytic cycles, is the fact that there are so many diverse methods. There are so many different mechanisms to pay attention to, and these different chemistries have different inherent selectivity patterns. So this can become quite bewildering to the practicing chemist. And so actually it was really Hugh who kind of motivated us to, and this was my good friend and colleague, former colleague, Tim Cernak, that we wrote this review article a couple of years ago now, and it's already, woefully out of date with all of the advances in the field. But what we argued for was this idea of a molecular toolbox for medicinal chemists to think about applying CH functionalization. And the basic idea was we could start to look at the different CH bonds in a molecule, and we could start to think about their inherent properties, such as perhaps their acidity, and perhaps use that as a means of selectively guiding functionalization to one place on a molecule. Or perhaps we could think about the nucleophilicity or the electron density at a particular carbon that we could harness for its reactivity. Or perhaps it would be the electrophilicity at a particular carbon that we could then engage some sort of nucleophilic radical in a Minisci kind of reaction. But then there are also the inherent CH bond strengths, perhaps at benzylic sites or perhaps adjacent to heteroatoms that would allow us to build in selectivity in a particular context. And lastly, as Jin was showing so elegantly, the role of directing groups. Perhaps there is a heterocycle that is nearby that can direct functionalization to a particular CH bond. And then, of course, we also have some catalytic chemistries that are really guided by the steric accessibility of certain CH bonds, such as CH borylation. So all of these concepts could be useful to help a medicinal chemist really think about their particular molecule and where they want to functionalize it and how they can go about achieving the desired result. So with that, I'm going to go into a few case studies. These will be short and sweet, but I hope they illustrate these concepts that I just discussed and give you an idea of how medicinal chemists would like to use these chemistries, where they are successful, and actually where some of the limitations are. And hopefully this can inspire folks who are out there devising new methods to think about what could make their methods even more useful and more applicable. So this first example comes from an adenosine 2A receptor antagonist program at Merck. And so Merck had a phase 3 candidate for Latinate, which was under investigation for a treatment for Parkinson's disease. And subsequent to that, we had a backup program that we were interested in some other therapeutic uses. And you can see that there was a change to the core of this molecule. So we had this fused tricyclic core. And so in the course of investigating the SAR, the team wanted to explore, obviously, lots of analogs. And in one aspect, they wanted to see about substituting this fused benzene ring. The challenge was that this anthranilic acid was the starting material for the entire synthesis, and it was a 10-step synthesis to make one analog. And so any substitution had to come in the very first step. So that's obviously very inefficient in terms of prosecuting SAR. But we wondered if we could use stage functionalization chemistry and perhaps get to the substitution patterns that we were interested in just 1 or 2 steps. And so, again, looking at this molecule, we saw that this CH bond was proximal to a heterocycle that perhaps could function as a directing group. And we also noted that this particular CH bond was the most sterically accessible of the sp2 CH bonds, perhaps could be open to CH borylation. And CH borylation is a chemistry that at Merck we have some history with. We had some ongoing collaborations with the Smith and Minisci group a number of years ago that really taught us how to practice this chemistry. And this is a chemistry we employ very regularly across many programs. Unfortunately, I only have time to show you one example today. So when we took this molecule and applied the borylation conditions, we actually looked at 2 classes of catalysts. We actually did some miniature high-throughput experimentation to really explore this more robustly. I'm just showing you the hits. In one case, we were able to find hits with some of these bidentate amine coordinated iridium catalysts that come out of the work of Hartwig as well as Smith and Minisci with this tetramethylphenanthroline ligand. These catalysts only have one vacant coordination site, so they are directed by steric accessibility of the CH bond. And so therefore we got very selective high yield CH borylation at this meta position. In the other case, we looked at these catalysts that have been reported by the Miura group. And these use these electron deficient phosphine ligands, and these tend to only bind 1 phosphine per iridium, therefore opening up an additional vacant coordination site. And therefore one can get directed borylation. And in this particular substrate, this just happened to be the perfect orientation. This was a very efficient reaction. Both of these reactions, we were able to scale up to gram scale to make these intermediates, and then we could do analogging off of those sites. And with a molecule of this size, we weren't necessarily looking to grow the molecules considerably, so we attached a lot of small groups. And I think that's one really important aspect of CH functionalization is having chemistries that can break the CH bond, install some kind of functional handles that can then lead to these small point edits that Jen was talking about earlier. So this was a very successful application of CH functionalization catalysis that really shortened the synthesis and really allowed the team to very quickly and robustly explore the SAR of this series. So moving on to another example, this is from a Bruton tyrosine kinase inhibitor program that we had for a number of years looking at applications in rheumatoid arthritis. And there's some very nice MedChem papers that were published from this effort. I point you to the references at the bottom. This particular lead series had a number of very favorable properties, and I'm showing you some of the key interactions here on this cartoon. We won't go through all of those now, but just to orient you to the molecule, it had a series of 4 linked rings. This hydrophobic A ring at the top was reaching back into a hydrophobic pocket. The B ring was more of a spacer. The C ring really made the key hinge binding interactions. And then the D ring occupied the ribose pocket. And so this molecule had excellent potency, selectivity, was quite clean on most off targets, with the exception of the potassium channel. The hERG had some activity that the team wanted to get rid of. And it had some non-optimal pharmacokinetics, so the clearance was a bit high and the bioavailability, especially in the rat, was low. So the team had an active effort ongoing, obviously, to address all these issues. And the question was whether or not we could apply CH functionalization chemistry to help them to accelerate their efforts. So looking at the chemistry about how you put this molecule together, there were a number of challenges that were, slowing down the efforts. On the A-ring side, this was something that could be assembled without too much trouble in 3 steps, this AB ring combination. The CF3 group had to be retained on this pyridine. It was very important to make the hydrophobic interaction. The issue was very limited availability of this type of building block. And of course, as Jeanette alluded to, we really want to use access diverse building blocks so we can really robustly and quickly explore SAR. But when you don't have the building blocks, that can be a real challenge. So the issue was that one of building block accessibility, but also this amide bond coupling was quite inefficient just due to the electron deficient nature of this pyridine. And so again, that was hampering the numbers of analogs that could really be robustly explored. Over on the CD ring side, this was essentially, this combination was built through condensation. And so again, the entire intact D ring was essentially coming in as one piece. And as you can see, that would really constrain the ability to do small edits and changes to this structure. And both the C and the D rings were suspected as being the metabolic soft spot. And so, the substitution patterns were set in this first step. And so again, this hampers one's ability to try to make fine edits to the molecule. So when we look at this from a CH functionalization perspective, we noted, of course, that we had some electrophilic carbon sites that we could potentially use Minisci chemistry on. And so that was definitely going to be something we wanted to explore. We hoped we could use some sp3 CH functionalization chemistry on this ribose pocket bearing D ring. Unfortunately, all of our efforts here failed, and so I won't really be talking about that. But I really wanted to highlight that because I just, think it's important to dwell for a moment on these are the kinds of challenges we face. And if people are out there looking for ideas of new tools, one really needs to think about this type of challenges, functionalizing a ring like this in situ without having to go back to de novo synthesis. So moving on to the story then of what we were able to accomplish with the team, applying Minisci chemistry to this A-ring, we first started with some conditions that were published by the Molander group back in 2011. And we've become quite fond of the Minisci reaction. It's generally something that applies very well, even in the context of a very complex molecule like this. The advantage of the Molander conditions is you give a lot of diversity of R groups that you can attach. So we're able to make some analogs, just showing you a couple here with some secondary alkyl radicals adding into this heterocycle. You can see that we lost a lot of activity, and so that showed that this probably wasn't going to be a fruitful means to explore. We also employed some conditions that have been developed in-house by my colleague Dan Duraco in the process chemistry department, and this was for installing methyl radicals. And med chemists love methyl groups. They're small, they're generally non-perturbing, and you can see that in this particular case, we got both methyl isomers, and this orthomethyl was somewhat tolerated at this position. But overall, even though we were able to help the team explore some of the SAR in this ring that they really couldn't get to otherwise, we could see that there wasn't a lot of to be gained by exploring further in that location. So we turned our attention to the C ring, and here it turned out that this wasn't nearly as electrophilic as we had thought. And so it turned out that in this case, that more electron deficient radicals were more matched to the inherent electronics of this ring. And so we're able to use this photoredox triggered Minisci reaction with alkyl iodide to install a number of alkyl groups. And one can see that a CFQ group was somewhat tolerated in terms of potency. And in parallel to this, the team was making a few analogs de novo, and they were able to show that these other small groups, such as methyl or fluoro, were well tolerated. So this was a very encouraging sign to the team, because this can potentially attenuate the electron richness of this ring and perhaps help with the PK. So what we really wanted to do was access the fluoro substituent in that position. And so we turned to electrophilic fluorination, which is another reaction that we've got some history with in our group. And we were able to do some screen on that, and we found that SelectFluor2 was the optimal fluorinating agent. There was an in situ trapping of methanol, and we were able to then eliminate to the desired fluorinated heterocycle in very good yield. And then we had a nice fluorinated building block C-D ring combo that we could then mix and match with A-B ring pieces to really explore robustly that change to the molecule. And one of the molecules that came out of it is shown here. And you can see that we retained potency very nicely. We've retained our off-target profile fairly nicely. Unfortunately, we weren't able to clean up the herd problem, which is most likely coming in from this D ring. And so it really points back to how we really wanted to try to tune the electronics of this ring, but weren't able to. But we did definitely make some improvements around the PK. So it just goes to show you how really employing these CH functionalization methods allows you to make these point mutations very rapidly to explore these different hypotheses. So for the last example I want to share with you this afternoon, I'm not able to share the program context, but this is the chemistry series that we have. This was an A's-benzimidazole series. This was one of the lead series for one of our medchem programs, and it had a lot of very favorable properties. Unfortunately, kind of late in the game, some issues cropped up that caused this series to be deprioritized despite all of its attractive features. One of those was PXR activation, which has to do with drug-drug interactions. So that was definitely something of concern. But of more concern was a bioactivation that was picked up in some of the exploratory safety studies. And it wasn't really clear what in the structure in the molecule was leading to that. However, there was a hypothesis that was generated from an X-ray structure of this molecule bound to the PXR that suggested that if we could substitute the 4 or 6 positions, we might be able to at least mitigate that liability to the molecule. But unfortunately, when the team was looking to explore this synthetically, they really struggled to try to generate analogs that had substitution at these 2 positions, and therefore reached out to our group to see if we could help with this. So what we did was we went back to that favorite platform of ours, the CH methylation reaction, and we went ahead and screened that, and we actually came up with quite a number of analogs. In fact, every activated position on these pyridines was functionalized with a methyl group, and many of the difunctionalized analogs were also generated. And so what we were able to do is really tap into the power of our separations group that we have at Merck, and most pharmaceutical companies have a very good separations group. And they were able to tease apart every single one of these analogs for us, and we were able to get structural identification and register these and get these into the assay. And we were really excited when we got the data back, because what we saw is that when we had a methyl group in the 6th position, we did not get trapping by glutathione in this microsomal GSH trapping study, which is essentially an in vitro surrogate for this bioactivation phenomenon that I had talked about earlier. Whereas if we only had a methyl group in the 4th position, then we still saw that glutathione trapping. So this really sort of put a spotlight on the 6th position as being the likely spot of glutathione trapping and bioactivation, and gave us a real mitigation path forward for how we could address this issue. And this could potentially resurrect this series for the team. So people were very excited about this, and they wanted large amounts of this compound very quickly in order to really test this hypothesis in an animal study. Unfortunately, as I just showed you, this particular reaction generates multiple compounds at once. So there's really no good way to use this reaction to generate gram quantities of this desired molecule. And as I also mentioned, the team really had struggled to try to make these molecules via de novo synthesis. So we had to kind of go back to the drawing board of CH functionalization and see if we could come up with another way of making this molecule. So taking a step back then and looking again at the inherent properties, we already exploited the Minisci chemistry to look at the electrophilic carbon. We can see that this pyridine can potentially function as a directing group, but that's not the position that we want to functionalize, the 4 position. We also noted that these alpha positions to the nitrogen, these pyridine rings, should also be somewhat acidic. And perhaps the desired 6th position would be the most acidic of all of them. And so this really led us to consider using some of the very elegant chemistry that has come out of Paul Mochel's labs, as well as others, using these hindered amide bases. He's shown that this chemistry can work very nicely in the context of complex molecules to install, for example, halogen atoms that can then be used for further diversification. So we had been interested in this chemistry for a while and actually had invested some effort in developing an automated strong base screen. And so we applied that screening method to this particular ABI lead. And we were quite happy to see that we were generating the desired iodinated product. So we saw both iodination at the 4th position as well as the 6th position. And what was even more exciting is that when we were able to add BF3 etherate as an additive, we could essentially flip the selectivity in favor of the desired 6-iodoisomer. By the way, this screening methodology has been published, so if anyone wants to see more details on how we do this screen, you can check out that reference. When we scaled this up, the reaction actually worked even better than it did in the screen. So you can see, which is this TMP magnesium chloride lithium chloride adduct, we saw a very nice conversion to the 4-iodoisomer. But when we added BF3 etherate first and then added the base, we saw almost a very exquisite selectivity for the desired 6-iodoisomer. So with that in hand, we actually went ahead and did the scale up. And what was really great about this, and getting back to this idea about speed, is the team, because they had been so interested in this molecule, they actually had profiled it extensively and they already had many grams on hand. So we didn't even need to go back and resynthesize this. We could just take the molecule we had in hand, apply the chemistry. We had 75% isolated yield on a 15-gram scale, working with our colleagues in the process chemistry group. And just to give you an idea of how we think this works, we think that the BF3 etherate is coordinating to this bottom pyridine, thereby blocking its ability to act as a directing group, and that's therefore opening up the possibility of the base-mediated deprotonation of the 6th position. A subsequent cross-coupling installed the methyl group, and we were able to deliver 6 grams to the team for their safety study in less than 3 weeks. So this was really enabling to the team in terms of putting this series back on track. So this was my last slide. I just wanted to come back to where we started with voriconazole and say, you know, that now that we have multiple ideas about how we can access different CH bonds, it really kind of gets to this ability to start to think about being able to take a molecule like this as is and start to tune where we can hit the molecule, the CH bond selectively. Sort of like Jen was describing a very beautiful catalyst-controlled example with quinoline. In this case, with voriconazole, we actually found with our strong base platform that that particular bond is the most acidic. And we can install an iodine there very nicely through the Noshel-Hauser bases. We can also exploit the electrophilic nature of this pyrimidine ring and do Minisci chemistry there selectively. And if we exploit the inherent electron density of this difluorobenzene ring, we can actually, under acid-mediated halogenation conditions, selectively brominate. So with 3 different sets of conditions, we can hit 3 different sites on 3 different rings of voriconazole. And of course, we're just getting started. There are many other CH bonds on voriconazole that one could envision being able to hit selectively. But I think as a field, we're making progress. And as we continue to make progress, we're going to see this being even more useful for medicinal chemists. And that just brings us back to this whole idea of the toolbox, which I'm really happy to see people in the center investing in and helping us fill it out. So with that, I'd like to just stop and thank the folks that did the work. This is our HCA team we have in Kettleworth, New Jersey, a great bunch of folks. I also really want to call out our analytical chemistry colleagues, without whom we would not get very far at all in terms of figuring out what we made. And of course, the purification group, I called out their work earlier. Our process chemistry colleagues have been really instrumental in helping us make progress in this area. And of course, all the discovery chemistry teams that we collaborate with. And with that, I'd like to thank you for your attention and happy to answer any questions.